Poseida Therapeutics today. We're a CAR T cell and gene therapy company. Been around for approximately 5 years at this point. A little bit of interesting history for the company that we are a spin out from the standpoint of the technology from a company called Transposon that's been around from the early 2000s. So it was Transposon, our founder, our CEO, Eric Ostertag, that developed this technology, particularly the piggyback transposon system, a non-viral transposon system or non-viral gene therapy system, as well as the Cas-CLOVER gene editing system, which found the basis of the company at University of Pennsylvania and subsequently developed that at Transposon as a toolkit, then spun Poseida out in approximately 2015 in order to create human therapeutics using these technologies. There's a number of other technologies have been brought into the company since. The company has grown quite rapidly over the last 5 years, up to over 100 employees now, and 4 products that are either in the clinic or near to being in the clinic, and a number of other products which are in preclinical evaluation for consideration to bring into the clinic. This is a little bit of what I was talking about here. This is our platform of gene therapy and gene editing technologies, quite significant, largely founded on the piggyback transposon system, which is a transposon, a non-viral gene insertion system. Talk about that a bit more detail in a few minutes, but also the Cas-CLOVER gene editing system, which is a unique and proprietary gene editing system. Number of other technologies you can see moving across the slide there, both in terms of the binders we use in our CAR T cells, the safety switches we use in our CAR T cells, and other gene therapy products, as well as different carriage systems such as AAV types and nanoparticles, which we have quite a bit of proprietary work that's being done or in the process of being done in. Let's see. So what are the products that we have right now, particularly from the CAR T space where we got our start in humans several years ago? We have a BCMA-targeted CAR T cell, PBCMA 101 for multiple myeloma, PSMA product, targeted product PSMA 101 we're looking to bring into the clinic in the immediate future to treat prostate cancer, a MUC1 targeted CAR T cell that should treat epithelial tumors broadly. So that includes pancreatic cancer, breast cancer, colorectal cancer, lung cancer, as well as an allogeneic form of our BCMA-targeted product, and potentially allogeneic forms of all these products. And that is founded on now the gene editing system for the first time being used in patients, as well as gene insertion platform, the Transposon system we've been using in patients for quite some time now. We also have a number of gene therapy products, straightforward gene therapy products for genetic diseases, things like sickle cell anemia, pediatric liver diseases like ornithine transcarbamylase deficiency, which we are looking to bring into the clinic, or I should say evaluating in preclinical studies now to decide what to bring into the clinic. This is a bit of an overview of how the transposon system works, the piggyback transposon system. So transposons are this here, which are a cargo, a gene, a transgene cassette flanked by ITRs, which are carried by a plasmid to be transfected into the cells or nucleoporated into the cells. That's accompanied by a transposase, an enzyme, which will then cut the DNA sequence of the transgene cassette out of the plasmid and insert it into cognate recognition sequences within the host DNA, so permanently transposing or transducing those cells. All of our CAR T products are based off this and have a very similar design, which is obviously the ITRs to make this transgene cassette, a binding domain, which in our case is generally a unique CAR binding domain, a safety switch which allows us to eliminate the CAR T cells or other gene therapy products should we want to, should there be particular adverse effects that we want to eliminate, and a selection gene which allows us to, during manufacturing, select out a pure product for administration to the patient, potentially increasing the therapeutic efficacy. And one of the key points to this is it highlights a number of advantages of the transposon system. one of which is the large cargo capacity. So the first thing we came into this with was large cargo capacity. We could put a large transgene cassette in. We could put in a safety switch. We could put in a car. We can put in multiple cars. We can put in a selection gene and more if we'd like to. There's a number of other advantages that came along with that as well, though, such as the TsDM phenotype or the phenotype of the cells we were creating when we produced CAR T cells that are unique to the Transposon system. So this is an illustration of that first advantage, so the cargo capacity of CAR T cells. As a proof of concept study here, we've taken that construct that I just showed you and inserted 4 different CARs in that construct. So made 4 different, a single cell CAR T cell product that carries 4 CARs in each cell targeting 4 different targets. And you can see that as we added each one of those targets from top to bottom, we gained the ability to kill cells that express that target sequentially. So proof of concept, and very early on gave us the enthusiasm for pursuing this in the clinic. However, it was the second finding that perhaps did even more of that and pushed this product forward and has continued to push it forward in the clinic. And that is the discovery that transposons produce a uniquely high percentage of TsDM, T stem cell memory, in a CAR T cell product. What are TsDM? So these are the T cells that are responsible for long-term immunity in the body. So they persist in the body for a long period of time. They can re-expand and produce daughter cells, effector cells, when re-challenged by an antigen. So these are the cells that, when you're immunized today against a virus, confer immunity to that virus 20 years down the road. We did not know this at the time when we first discovered this, but since then, a large number of other well-known institutions have published data indicating that the percentage of TsDM or CM in a product correlates strongly with efficacy of that product, of that CAR T cell product. And that includes us, so our CAR TCR presentation from early last month is one of those listed above, but I think you'll recognize a number of other CAR T cell companies and organizations and academic institutions that have published on this topic. And this is the data that backs it up. So this is left-hand side and right-hand side of this graph, the same product produced with either a lentivirus vector on the right-hand side or piggyback transposons on the left-hand side. And you can see the significant higher concentrations, significant higher percentages of TsDM and CM on the left-hand side versus the right-hand side. The first question we're going to be asked, of course, in the follow-up session is, what is the molecular mechanism of this? We're working at, we have not elucidated this yet. There are some evolutionary reasons why this might happen, and it makes sense for a transposon to do this. But it was a very unique finding when we first saw this, and particularly interesting that as we progress through the clinic, the correlates have continued to fall into place. So now I'll get into a little bit of a focus on the specific products we have, what data we have for them thus far, and why they're particularly interesting. From the earlier summer I was talking about, we have BCMA products for hematologic malignancies. That's specifically multiple myeloma. We have a PSMA and a MUC1 targeted product for solid tumors. That's prostate cancer and solid tumors broadly. And allogeneic version of currently the BCMA targeted product, but potentially all of the products down the road. Starting out with a brief overview of PBCMA-101, the BCMA targeted product for multiple myeloma. This is the very interesting graph which led us down the TsDM routes 4 or 5 years ago. So we had produced our first product, PBCMA-101. We had created CAR T cells from that from healthy volunteer donors at the time. These are, in the clinic, autologous CAR T cells, so they come from the individual patient. But at this time, we had initially produced them from healthy volunteers. We tested them in our first animal model, MM1S model of multiple myeloma, and we saw something quite unique. So you can see the untreated control animals here, where the tumor just grows continuously until the animals are sacrificed. And you can see the PBCMA-101 treated animals here, where the tumor grows up, the CAR T cells are administered, and the tumor is rapidly regressed. This is very typical for what you see in a CAR T cell product. The tumors then remained in regression for a period of time and then began to regrow, also what you see for many CAR T cell products. What has previously been reported in this model from both these investigators and others is that this will continue to grow. These tumors will then follow the same course as the untreated control animals. However, in this case, these tumors spontaneously re-responded, the T cells re-expanded and re-eliminated the tumors, and they remained undetectable or nearly undetectable to the end of the experiment. So that's what originally got us looking into the TsDM, the correlation with the TsDM phenotype with the product. And of course, made us very enthused to take this product into the clinic as quickly as possible. So we designed a fairly typical phase 1 oncology or CAR T cell study. That study received a grant from CIRM, the California Institute of Regenerative Medicine, which paid for a good part of the conduct of the study when we were an early stage biotech company, so that was extraordinarily useful. But very typical 3+3 design, very typical Cy/Flu conditioning regimen as you see for CAR T studies. Some of the unique aspects of the study are that we allow for outpatient administration. That was based on the early findings, good safety profile of the product, which I'll talk about in a few minutes, and an amendment with the FDA for an allowance for all outpatient dosing, which we do frequently now. Also amendments along the way to allow redosing of patient with CAR T cells. We don't do that as much as we do outpatient dosing, but it's a very unique potential for a CAR T cell to be, for a patient to be re-treated with a CAR T cell after failure of either another CAR T cell or the same CAR T cell. And that's something we're very interested in investigating right now on both angles. What happens if you treat a patient who has failed CAR T cell A with CAR T cell B, and what happens if you treat a patient who has failed CAR T cell B with CAR T cell B. We've initiated a pivotal phase 2 trial since then based on the findings in the phase 1 trial that I just spoke of. Very similar design to the phase 1 trial, outpatient dosing. Of course, in this case, no allowance for retreatment, but a limited dose range and 100 subjects, which modeled off another number of other CAR T cell studies, is expected to be potentially registrational. Large number of institutions participating in the study that you might recognize up there. A lot of enthusiasm amongst investigators and patients, given the early data. This product also received RMAT status approximately a year ago, and I think that has also furthered the continued development of the program, of the product. This is the last set of data we published. You can see, as you would expect with CAR T cell product, very high response rates. An example of one of those patients on the right-hand side where the patient had visible plasmacytomas pre-treatment, i.e. visible tumors on PET pre-treatment. Those tumors resolved after treatment with PBCMA-101. The safety profile, quite unique, and we believe also potentially linked to the TsDM phenotype of this product. So you can see that the overall CRS rates are quite low, somewhat of a dose response, albeit not an extraordinary dose response there on the right-hand side. But the overall rate, which has wandered around 10 to 20%, is quite low across the board when you look at other CAR T products, not only from the standpoint of the rate, but also the severity, and the highest severity has been grade 2 at this point. And there's only been one case of neurotoxicity or suspected neurotoxicity at this time in a patient that was treated with PBCMA-101, had confusion at baseline, confusion worsened, then improved on study. I don't have the slide here, but the linkage or potential linkage between the TsDM and that safety profile is that when you look at some of the other presentations, the longer presentations we've had that focused on the clinical data, you'll see that the expansion of these CAR T cells is more gradual than what you see with most products. So instead of peaking, having an expansion peak in patients of perhaps 5 days or 7 days, these peak at 2 to 3 weeks. So we think that gives the patient a much longer time to adjust to the ramping up levels of cytokines being released from the CAR T cells, and produces overall lower peak levels of cytokines because the release into the system is spread out over time versus simply administering a large amount of effector cells, which immediately go in and attack the tumor cells and kill the tumor cells, but release high peak levels of cytokines immediately. Moving on to the second product, so the PSMA targeted product, also quite interesting. Same construct we talked about earlier, a centurion, a novel binder, novel type of binder instead of an SCFV that targets PSMA, highly expressed on prostate cancer cells, as you might expect. You can see the mice here, similar experiment to what we did with BCMA, let LN-CAP tumors grow up, administered the CAR T cells. Similar curve, but unique in a way, in that you have the tumors grow up, administer the CAR T cells, the tumors resolve, and stay eliminated until the end of the experiment. You don't see that double hump here, and we think the reason for that is because the kinetics of response of a solid tumor are different than those of a hematologic tumor, and particularly multiple myeloma. So longer time to response, the second hump, regrowth, et cetera, may be buried in there. But nonetheless, you see what I was talking about earlier in terms of the cells you see circulating in the mouse blood during this experiment. So this is the typical phenotyping of CAR T cells for the TsDM and the various other effector and central memory types. TsDM, the TsDM phenotype is located in this quadrant. You can see in mice that are bearing the tumor, you start out with TsDM, these differentiate into effectors, tumor's gone, they redifferentiate into TsDM and lie waiting in blood, in bone marrow, to be a re-exposed antigen, to be re-exposed to tumor. In the top set of panels, you see that the cells remain in TsDM phenotype throughout because there's no tumor in this control group, no antigen to be exposed to, nothing to differentiate them into effector cells. Moving on to our Allogenic program. So in this case, we now utilize that same base transgene cassette to create CAR T cells, but we edit them with our Cas-Plover gene editing system, double knockout of beta-2 microglobin and TCR to eliminate alloreactivity, both graft versus host and host versus graft. You can see we've done that here. These are the knockouts. This is elimination of alloreactivity in both directions. The cells still bear the TsDM phenotype, and that is likely in part secondary to the uniqueness of the Cas-Plover system and the ability to edit resting T cells. And you can see the efficacy on the right-hand panel here, equal in quality to killing target cells to the initial product, the PBCMA-101 non-edited autologous product. So here's a summary here. Very broad platform in the company, a lot of uses, a lot of things we've used it for, particularly CAR T cells to start with, number of advantages in those CAR T cells in terms of efficacy, safety, the TsDM phenotype, the binders, now expanding into solid tumors where the TsDM phenotype may again play a significant role in terms of exposure to wave after wave of effector CAR T cells that could potentially eat through a solid tumor, which is not seen with the liquid tumor. And then finally, the unique aspects of the gene editing platform that could bring us very nicely into the allogenic field. And that's all I have.